Hello. Uh, Welcome to NICO's Wednesday at NICO seminar series for the new quarter and new academic year. Before I introduce today's speaker, I just want to put in a plug for the rest of our seminar series listed up here on the right, Wednesdays throughout the quarter. We've got some great speakers coming. And also to mention data science nights, which are starting tomorrow night here in this building, Chambers Hall at 5:30. And we've got a few different nights lined up. You can see here on the future dates for the quarter if you've ever been curious about what people do at these nights, or you have your own data set you'd like to get other people's input on or learn some new tricks for dealing with data. It's a great opportunity to connect. So please come by for the data science nights. And you already made the right move coming by for today's seminar. Come by for the rest of them also. With that said, I would like to just introduce today's speaker, Emma Alexander. I still think of her as new faculty here in computer science, but you're not that new now. Newish,
Yeah.
here a few years. Emma came to Northwestern after doing degrees in physics and computer science as an undergrad at Yale, which maybe is why you mentioned physics-inspired methods, I think, in some of your work. And then PhD from Harvard, postdoc at Berkeley, great CV there. She works on computer vision, which I have discovered due to my ignorance. I didn't know it was such a hard problem. Actually, I've been digging into it a little bit. We'll have to talk another time about this in my own research. I also know you've done work on jumping spiders, which is awesome. I know they have some amazing vision system. But Emma's also been a member of the National Academies of Engineering Frontiers of Engineering program, which I did years ago, but very prestigious invitation that you got to that. And I saw you listed Fulbright also. Were you actually a Fulbright scholar?
I did a specialist program, so it's a shorter version. Yes.
That's great. Where did you go?
I went to Jabalpur, India.
Amazing, amazing. Okay, so I'd love to chat with Emma all day, but to let everybody hear about her research, I'll let you take over. Emma, welcome.
Thank you. Hello, everyone. Welcome for showing up right as we're all sort of dizzy with the start of term. It is an honor to be the justification for your free taco this afternoon. I'm going to be talking today about constrained information optimization in visual cortex. But before that, I just want to introduce my lab, the Bio-Inspired Vision Lab here in McCormick. The field that we are in is computational imaging. So just as a very, very high-level overview, what computational imaging as a field seeks to do is to interpret light as it moves through scenes into optics and detection through a computational pipeline to get scene information. This is a way that we can sense a lot of information about the world. What makes this computational imaging as opposed to, say, mainstream computer vision is that we co-design the optics, the sensor, and the computation. And this is, of course, going to be in the service of specific information that we care about in the scene. So this is really exciting, okay? This allows us to see stuff that we couldn't otherwise see, including things like cells that are unstained, living, and functional. You can see into a human body without harming it. You can even look at physical phenomena like shock waves or the black holes at centers of galaxies, right? So all of these images we couldn't get from algorithms alone. We also have to change how we detect them. So there's a lot of really exciting, vibrant, cutting-edge work being done in this field. I would also argue that this is the normal way for vision to work, and the computer vision, which only develops algorithms, is the kind of odd man out. That's because vision has existed as a capacity for about 500 million years. It's happening all around you, all the time, and all of these systems have optics and computation that developed in tandem in service of specific scene information. Okay? So there's a whole bunch of computational cameras that exist out in the world, and there are technological reasons to care about them. So we think a lot in computer science and in engineering about the state of the art. And biology, particularly in vision, is very far beyond the state of the art, and not just in the metrics that we know how to benchmark. It gets a lot of these like very hard sort of qualitatively different aspects in their overall system performance that we're not yet capturing as a field. So first of all of these computational cameras, I'm just going to point out there's a ton of design diversity here. Once you open up the optical and sensor design, you have a ton of degrees of freedom. It's really hard to even know where to start. 500 million years of optimization have at least shown us some locally good solutions to some problems. All of these systems are also compact. They exist within a single organism. They run in real time, right? If you see a predator too late, no, you didn't. They're very robust. We don't see them making the sorts of foolish, brittle errors that I think we're all becoming a little more familiar with in the AI years. And perhaps another critical issue that's being raised now is they're all low power, right? These are animals that acquire calories at risk to their lives. You can't just plug them into a wall socket, let alone a data center that is going to run up the power bills of everyone in the Chicago area, right? So this is a different way of doing vision. And I would argue that it has these qualitatively different characteristics that we need to embody as we develop new visual technologies. So here's an example of some of the animals that we've thought about in my lab. We thought a lot about how jumping spiders use defocus. I'm not going to talk about that today, but that is my favorite spider. I will talk about her forever. We think about how fish see motion. We're looking at a number of different computational ways of handling color across a range of species. And today I'm going to be talking about some projects we've done on stereo 3D vision in monkeys and orientation sensitivity in mice. I'm not going to be talking about any specific cameras today, so we can abstract this to really just be about the information processing principles that they're using, right? It's kind of application agnostic. Okay, so what we're trying to do is not just the forward engineering computational imaging where we apply computing and physics principles to build cameras and displays. My lab does this. We make new algorithms. We build new optical systems. We make new kinds of cameras. But we also recognize that there are solutions in the world that we need to reverse engineer to uncover new principles, not just in biology, but also they can inform new principles in how we think about the computing and the relevant physics. And the cool thing about having both of these directions, right, is every time you have a new principle, you can build a new camera. Every time you build a new camera, you find a gap in your theory, right? And we can sort of walk around this circle over and over and over. What I'm going to talk about today is some very, very early days preliminary reverse engineering of figuring out how to handle brain data. So we're only going to see the blue arrow today. All right. So the question that I want to ask is, what are brains doing, right? They're out here, they're solving all these problems, they're doing it way better than we know how to do. It would be amazing if I could just rock up to an animal, be like, man, you're really good at seeing color, or identifying objects in the dark, or moving really quickly, or imagining a scene based on a couple of words of a text prompt. And what would be really cool is if I could somehow have an algorithm extractor that could look into the brain of this animal and like pull out the algorithm that it's doing. I cannot begin to explain how impossible this is right now. And the problem is that what are brains doing is a giant question, right? The short answer is everything that you have ever done or thought or felt or experienced, consciously or unconsciously, right? All of living is brain involved in some degree. So really what the brain does is it's a tool for accomplishing survival. And breaking that down into sub-goals all the way into how are you detecting color? There's a lot of work there, right? At the same time, brains are not computers. They also have to stay alive, right? They have to maintain health and function. So instead of being a bunch of kind of nicely characterized transistors that you've manufactured with all of these high precision processes, the computational substrate here is 100 billion tiny little wet salty bags that zap each other, okay? They have wet, salty bag business that they have to attend to, or they're not gonna be able to keep zapping each other. And so if you don't account for that, you're gonna really misinterpret your data. It's gonna be a huge confound. So what we wanna get at is the heart of the information processing that happens in the brain. But in order to interpret that correctly and to be able to extract anything of value, we have to know how these factors of goals and constraints play into the things that we can actually observe, okay? So that's the big question for today. And we're going to think about this mathematically in a constrained optimization framework. So the perhaps bold hypothesis here is that we can think about the brain as being a maximizer of goals subject to some kind of need. I think that's not so controversial, perhaps. In order to make this at all tractable, we're going to pull in a few more specifics because what we care about is this information processing. So when I talk about goals, I'm going to think about the expected merit function on some information subject to biophysical constraints. So today we're going to dig into what this merit function might be and how to tell what it is. We're also going to think about how to model these biophysical constraints. But before I get started, I just want to clarify what I mean by information. I'm going to be using a Fisher information framework. So Fisher information quantifies how much a response can tell you about a stimulus. It tells you about the decoding potential of your system. We can define this as the variance of the score or the expected value of observed information. If that already makes sense to you and means something, take a beat. If it doesn't, I'm going to show you what it looks like pretty concretely for our problem. So I have a stimulus and a response and some curve that maps stimulus to response, right? So the stimulus, for example, could be the orientation of an edge in the world, and the response could be the mean firing rate of a cell in your brain, okay? So this would be a cell that doesn't care about vertical edges and starts to care more and more as you orient that edge, okay? So if I observe a response in the brain, right? If I sit there and I measure the spike rate, how well can I decode that, right? How closely can I tell that this is a horizontal bar? Or maybe it's a little bit off horizontal in either direction, right? So this is going to be a sensitivity. And if we call this the tuning curve H of the stimulus S, you could probably guess that this Fisher information, this sensitivity, it's going to be a function of the stimulus value, right? I'll be more sensitive to some things in the world than others. And it should probably have something to do with my derivative here, right? Because I want to see how much I can change before I notice a difference. Turns out the relationship is it's the derivative squared. But it's a little more complicated than that because this system, like all systems, and in fact, the brain more so than our standard computers, has noise in it. So I don't get a single response. Instead, I'm going to get this distribution of responses over time, right? So now, if I sort of abuse the notation of an error bar and scoot that over here, I have this tuning curve where I not only have a mean response that changes, but I also have a variance at every point. And it's standard for neurons to model them as being sort of having a Poisson noise distribution. So if I'm not firing much, I don't have a ton of noise. If I fire a lot, I have more noise. And in that case, you need to correct your sensitivity. And for a Poisson, you just end up dividing out by that tuning curve value. Okay, so this is somehow going to tell me I get some noisy measurement up here. How much can I tell about the x-axis from this noisy estimate on the y-axis? Okay, great. So the downside of this is that it's very local, right? It's not going to capture a global encoding. But the pros are significant. The first is that we can measure tuning curves. And there's not a whole lot about the brain that we can measure super easily. So we love just a tractable quantity we can work with. And once we can measure it, we can connect it to things that we care about. So for instance, you can use it to approximate the mutual information, which gives you this global property, but is extremely difficult to measure on its own. And you can also use it to measure discriminability. So if you've ever seen a claim like, oh, humans can see a million colors, right? There's a million things you can discriminate against. That's based on the Fisher information of your cone cells, okay? So how many things can you tell apart? Where are they? It's kind of like a fancy signal to noise. The top is signal, the bottom's noise. Fisher information. Okay, so what does that look like when we go from one neuron to an entire population of neurons? There's a lot of neurons in the brain. We're not sure what they're doing, but it's highly unlikely that anything important to you gets encoded in a single neuron. So we think about these populations as encoding useful information. So instead of just one tuning curve, I now have a whole family of them. And if I make some reasonable assumptions about their independence, I can just add up the Fisher information from each one. Great. Okay, so in real neurons, I'm going to have some kind of gnarly data. I'm going to have to estimate these tuning curves. I'm going to have to add them together. What's nice about this is that once you have a smooth enough curve and you can trust your fit a little bit, you can compute this Fisher information for any tuning curve, right? It's derivatives and divisions. It's not hard. Going the other way is tricky, right? If we want to say, okay, what should my cells look like? What would I predict? What does an optimal population look like? For that, we have to make some simplifying assumptions. So a lot of times what people will do is they'll say something like, okay, I'm going to start with a family of Gabors, right? I'm not going to let everything vary at once because it becomes too difficult to analyze. And then they'll sort of warp this in various ways. So a really common way to do this, which I'll touch on, is you can change the overall gain, right? So there's this envelope of your max firing rate per cell. And then we also change the width and how close they are together, which we can encode in a density function, okay? So how does this play into our framework? So this gives us the information measurement that we're going to work with. And now we need to get back to thinking about what's going on around it. So expectation of merit. First off, expectation. We live in the world, and our visual diet changes what a successful visual system looks like. So for this, we need to turn to what are called natural scene statistics. So you don't see, for example, horizontal lines as often as 10-degree lines or 20-degree lines. We're going to use stereo disparity as a variable for this section of the talk. So this is the relative depth from the target of the thing that you're looking at in the world. If you have seen a 3D image, the shift between the red and the blue is the disparity, right? So if it's what you're sort of looking at, they're going to be right on top of each other, disparity 0. If it's one direction, they'll shift this way. Other direction, they'll shift this way. Okay, and we can measure the visual diet of stereo disparity for a person. You mount eye trackers and 3D cameras on a person's head, and you have them go around in the world, and you can just measure what happens most often. And it turns out that disparity has sort of this characteristic natural scene statistic where most of what you're seeing, particularly near your fovea, you're mostly looking at stuff. So the disparity tends to be 0, and then it falls off near and far. That's for a task like walking outside. If you're doing closer work, like making a sandwich, you can have a sort of systematic shifts in this distribution. But there's basically any way you want to characterize this, there's always going to be a peak at 0. And that peak at 0 is going to be a very important signal for us. Okay. All right. So we have an expectation. We have to assume some probability distribution of a stimulus. Yeah. Mm-hmm. Yes. Yeah, that's an excellent question. So yes, they're going to have different geometry in their visual system. They might have systematically different eye motions, and they are also going to be in a different environment doing different tasks. So, I mean, if you really needed to get it right, that's enough factors that I would just go ahead and measure it.
Not something that people have studied. Mostly people have.
Yes.
When you are focusing on something.
Yeah. So to my knowledge, so this was only 10 years ago. I'm not sure that anyone has actually like gone out and measured this distribution for any other animal. Yeah, no, it's a good question. Different animals have different binocular. If you're really not using your binocular overlap, this statistic probably doesn't matter to you for what it's worth. We might not expect it to show up in any codes. It's a great question. Okay, awesome. Yeah, thank you. So we have some visual diet that corresponds to not just the world, but in fact our eye geometry and our behavioral needs and the environment that we're in. Now we want to think about merit, okay? What is it that we actually want to do with this information? And there's going to be sort of two common hypotheses for this. A lot of times people will talk about the infomax and the diskermax objective functions. So infomax, I told you before that you can use Fisher information to approximate mutual information, which is basically just a measurement of how much of the world did you get into your brain. A pretty good goal for a sensory system, right? That's arguably what sensing is. And we can approximate mutual information by using the log of Fisher information as our merit function. You can read all about the assumption. Oops, this is the wrong year, I'm sorry. I think that was early 2000s. 2013 maybe? Anyway, a lot of assumptions that go into how tight this approximation is, but again, it brings us from something that we can measure to something that's hard to measure, but we care about a lot. Diskermax says, I want to be able to distinguish between the two closest stimuli that I can. To do this optimization, you want to maximize the expectation of -1 over the Fisher information. This comes from the Kramer-Rao bound. You can think of this as your L2 or mean squared error loss, and you can actually generalize this to any LP norm loss. So if you care about the cubed error, again, that's going to show up as a polynomial in the Fisher information that you optimize. Okay? So, okay, which do we care about? It's not going to be uniform across the entire brain, right? We're doing a lot of stuff, but there's a general hypothesis of hierarchical computation, which as a computer scientist, like, is sort of this like, yeah, of course, like literally how else would it be? But the idea here is that you want to be general early on in your visual system. So you're going to start out looking like this infomax. I got to get the world into my brain. But then by the time you get down to specific tasks later in your visual processing, you're going to care more about things like what was my squared error, right? So this is, I got to know where stuff is in the world. And this is, I have to plan a hand movement through some thorns, and I need to know exactly what that gap is and not get it wrong. Okay, so I said early and late. What does that mean? We got to talk a little bit about the visual pathway. This is in quotation marks because I'm going to make an almost criminal oversimplification of how vision works in your brain. But if you've never thought about it before, here's a useful picture to have. So you start out, photons hit your retina. There's compute that happens in the retina. You send it down the optic nerve. You hit the back of your head. Okay, that's the first visual area, V1. V1 is pretty cool. Here's a stimulus that was shown to a monkey, and here is the stained brain of that monkey after it looked at the stimulus. You basically trace out the world inside your head. You then compute as you move forward, and you go into more sort of complicated and abstract quantities as you move forward, V1, V2, V3, et cetera. Okay, so this is a huge oversimplification. There's a ton of interconnection. There's a ton of recurrence. It's very, very complicated. But if you've never thought about the visual system before, V1 comes first, back of your head, move forward, get more complicated. We're going to be looking at a comparison between V1 and also V2, which is quite close and similar, and V5, which is deeper in the brain. It's this little red area, and it's going to encode 3D motion. Okay? What's cool about these visual areas is that one of the things that happens with this hierarchical computation is that typically the type of feature that you're computing on changes so extremely that it's difficult to have a direct area-to-area comparison. But V1, V2, and V5 all encode stereo disparity. And so we can put this information on equal footing and see what the difference are in how they're computing on this. Okay, so we got some data from a monkey lab. And this was about 1,000 tuning curves that they measured. So again, if I take a tuning curve, right, I get down to the Fisher information from each by taking that derivative, dividing by the noise. So my Gaussian tuning curve gets this sort of two-lobed Fisher information. I do that for all of my tuning curves. I sum them up in each area, and then I can have a reasonable guess for the Fisher information with respect to stereo in each of these three brain regions. Before I show you what that looks like, I'm going to show you a model that would predict how it should look. So remember that stereo disparity has this peak at 0 in its probability. The standard signature of optimality, like if you see that phrase. Usually what that means is that people have discovered that the information encoding in a region goes as p squared, which is the standard prediction if you put in the InfoMax objective. Okay, so if you're maximizing mutual information, if you're just trying to get your outside correlated with your inside, you should concentrate, you should be good at seeing stuff that is more common. Okay? If, on the other hand, you need to be able to tell the difference between two stimuli, now all of a sudden you've got to divide yourself a little bit more fairly across the stimulus space. And the standard prediction for this under a variety of assumptions is that your Fisher information should go as your root probability. Okay, so in any case, you should be good at looking at stuff that's going to happen more often, but their relative weighting is going to change. The hierarchical computation hypothesis said, early visual area should look peaky, later visual area should look flatter. And in fact, we have the first sort of quantitative characterization of how that change has occurred in a real brain. Something that's a little bit interesting is that the difference in these exponents is not actually, it's not 2 and it's not 0.5. So depending on which version of the, which task stereo disparity probability you use, you can get a variety of different answers. And all of them we have the right, or the expected trend, but in none of them is the difference as large as we would expect. This might be because V5 is still early on. It might be because there is a constraint that is pulling them away from the solution. It might be because of kind of any number of factors. But it is exciting because it gives us at least the beginning of a tool that says, hey, the computational goals between these areas is different, and here's the relationship. Yeah. Yeah.
When you said early on and later, early on be general, later be specific, I guess I interpret that as development, like over the life of an animal.
Okay.
Maybe you're going to get to this. I don't know.
Thank you for bringing that up. That I should have clarified. I meant early, late in the computation on the same piece of information.
So V1 versus V5, you're saying maybe early versus late.
Early.
Maybe it's not late enough in the hierarchy. But what do you think about, have you thought about this in the context of development?
That is a really great question. I haven't. Yeah, it's certainly true that the visual diet of infants is different. They have very bad vision, and it's not an optical problem, it's a brain problem. But I've never studied, I would be interested to see kind of what the developmental pathway looks like for this information. I don't know that that's been studied. That's a great question. Yeah. Anyone else while we're at this moment? Yeah.
So this is a very interesting conversation because Danny and I went on actually completely different directions with early and late, and neither of us was thinking of the one that you said. I was actually thinking about task switching, right? You are doing something in which you try to see what's going on in the world, and now you are doing something specific, and then it's when you get more kind of in this way.
Yeah.
And so from that perspective, it's really interesting that these exponents seem to be going completely the different way that I would expect, right? Because food preparation would be something that is more specific. So I would be thinking that the exponents in there would be lower, and that walking outside would be higher. You are talking about ear, and you are talking about earlier and later as being on the computation side. But when you are thinking about the different tasks, is the difference in the exponents the more significant thing? Is there anything to the actual values of the exponents?
Yeah, that is a really great question. So I think yeah, there is something like the need to have fine-grained depth resolution much lower when you're just taking a walk than when you're putting stuff on top of each other. I completely agree with that. You know, it could be that, yeah, I think maybe that supports this hypothesis that we're just still pretty far away from the end effector. But it's a very interesting question, and I hadn't thought about that before. I had just thought of this as, oh, there's a whole family of probability distributions, but that's worth some deeper thought. Thank you.
Just going back to Danny's question. So it turns out that in the first 3 months, infants don't rely on their visual cortex orientation. They rely on their midbrain. And myself and a collaborator have related this to the fact that they're essentially like fish, and fish rely on their midbrain in water because they're limbless creatures and they're being passively carried around just as infants are. And it's after 3 months that the visual cortex starts to take over. So there's some interesting stuff going on there, but no reliance on the cortex for infants.
Oh, I love that. Thank you, Malcolm. That's delightful. Any other questions? Any other interpretations of early versus late? I meant you start in the visual system, you get later in the visual system, but this is very interesting, yeah. Yes, okay, excellent, fantastic. Yes, yes, yeah, and I think this ties a little bit to like, yeah, what is your actual need behaviorally? Do you need fine-grained detail? Do you need to know about it right away? Probability is a thing that we can measure with cameras and sending people out into the world. I think you could argue that a better measure would capture more things, right? It would be like an importance weighting that wasn't strictly how frequently things happen. That is very difficult to model. But yes, I completely agree. Yeah.
I mean, if you want to prepare for the food,
Yeah.
but you want to grab a tomato, but you failed.
Yeah.
So would you like to measure that kind of thing? What happens in the brain?
It's a good question. So one of the things that that methods like this really fail to capture is that there is feedback, right? If you're grabbing, if you're working in the world, one of the biggest sort of crimes against the visual pathway that was on that slide is that I just presented it like, oh, it just moves forward, like it's all talking to each other. So we have not thought about anything that high level, right? This is strictly if you are with standard adult human competence walking outside or preparing a sandwich. This just happens to be the information that comes into your, really, this is at like a retinal level, right? It's just the information that's in the world available to you, which we're using as an approximation of what's important. But yeah, you guys are right. All of this stuff is important and not yet modeled. Yeah.
I have another one, which is also an inspiration.
Yeah.
So another way to think about the longitudinal is about from an evolution perspective, like 500 years ago, 1,000 years ago, how would those kind of like pattern changing, and how would like number or density of information when you're processing a number of tests happen at the same time, like impacting those processes?
Yeah, it's a great question. And there's been some study of like plasticity in animals where we have some evidence for when the evolutionary history matters versus when the life history of that animal matters. You can, like, for example, make a cat really bad at seeing vertical lines if they never see vertical lines as a kitten. So it's a mix. And there's probably a lot of variety in there. I am gonna pop into our other half of the question though, because I think there's other cool stuff you guys want to know about. Okay, so we thought a lot about the expectation of merit, and we saw that there was a signature that what the merit function was in these two areas of the brain was different, and that this supported a wider idea that there's a systematic change in how you need to handle information as you move through the brain. This all assumes that the constraints didn't change, because for a constrained optimization problem, the objective and the constraint hold equal weight. They can both change your solution, right? I think it's reasonable to say that V1 and V5 are probably not hugely different in what constrains them, but it did get us thinking about when a change in constraint would matter. Okay, so for this, we're gonna move into some data from mice. Okay, so how should we think about constraints? This is an area where we do kind of have to go to these cartoon simplified neural populations, but then there's some pretty cool math on how to optimize populations like this under constraints. So we make some assumptions about the shape, and then people will constrain stuff like the number of cells that you have, their mean firing rate, their max firing rate. Sometimes they'll directly constrain the information, right? There's a bunch of different things that you can constrain. And at any given state, like, sure, these are all finite. You could constrain them. But I think what makes a good constraint is not just that it happens to be true and gives you an answer, but that when you turn the dial, you get a change that is correct, right? You shouldn't just capture one single state. You should be able to vary the constraint and see a real change that occurs in the world. So for example, I'm going to take a tuning curve that really likes horizontal lines. One way that people will constrain this tuning curve is they'll say, oh, the mean firing rate is like the energy. That makes sense, right? Like, spikes are expensive, send fewer spikes around. And what you do there is you just end up shrinking the whole curve. Otherwise, you could say, oh, we actually want to keep this, this max firing rate the same, but reduce the information. That ends up widening these curves. So there are now at least two distinct hypotheses for what should happen when you tighten up the constraint on a neuron. And we wanted to figure out, like, is this right? What's the thing that we should be using to constrain these population codes? So we wanted to really zero in on energy, because this is a constraint that really matters and it changes over time, right? An animal that can only perform well when it's well fed will not last very long. And so we think that probably these population codes are not just optimized for a single energy state, but for a whole range of states, for a robustness to energy changes. Okay, and there was recently some new data that came out on what happens when you put a mouse on a diet. Its visual system will change. So they put these animals on a calorie deprivation regime for 2 to 3 weeks. They lost 15% of their body weight. And amazingly, when they go into the visual neurons, they saw that they were using about 1/3 less energy than they did before. Okay, the kind of crazy thing was that they were still firing the same amount, which is a little bit strange, right? What's cool about this, and again, what's exciting as a technologist for what happens here, is that they experienced a graceful failure in visual function. So they didn't just get hungry and go blind, right? Which is how our devices currently work, right? You're taking video, maybe in like a personal safety device kind of context, your battery's low, right? I'm stressed out just thinking about it. You're gonna take these beautiful crisp frames and then your camera's just gonna die, which is not ideal and not how any animal has ever worked. So what I mean by graceful failure is the mouse was shown a pair of oriented stimuli, and it had to go to the correct one to get its little reward, right? And so this, you can see, easy to tell the target and non-target apart. As the non-target gets closer and closer, it's going to be harder to tell it apart. And what they found is that in black, when they're well fed, okay, the closer the target get, you sort of gradually fall off. But what's interesting is that when they restricted the food, you have this sort of cave in. So if you're 10 degrees or less different, all of a sudden you start failing, but you maintain your coarse detail, your big picture, right? So what this is suggesting is that you have sort of this like wave of gradual failure where you're not just on and then off. Okay, this is very exciting. We definitely want to know how to do this with our technologies. And when you look into the codes of their cells, the tuning curves are neither just shortening or just widening. They're doing both. They're flattening, and they're flattening specifically in the way that allows them to maintain these firing rates. Okay? So I'm going to give you a just so story here. Firing is less expensive than you'd think. Most of what's expensive is moving ions around. You want to reduce the amount of current that your cell uses. The way to reduce current, according to Ohm's law, you can drop the voltage change that you need, or you can mess around with either the resistance R or the conductance G, which is just its inverse. Okay, so typically what a cell does is it sits at a resting potential. As it gets in signals, it comes to a firing threshold and then spikes. If you bring the resting potential up, you don't have to move as many ions back and forth. But now you're sitting closer to threshold, you're more sensitive to noise, so it's cheaper to send and receive signals, but you're going to be more vulnerable to these false firings. There's this Poisson noise where these false firings are positive and not negative, and so you end up boosting your signal at the edges. To maintain homeostasis, you have to pull down the gain of your peak. What this ends up looking like is that this homeostasis is really, really important. Okay, that was the surprise from the data. These brains really fight to keep sending the same spike rates as they were sending before. So when the spikes become more expensive, they just use cheaper and worse spikes. Okay? So our hypothesis is that these cell changes might be information optimal in the noise energy trade-off, right? So there's a bunch of different ways to be cheaper. I want to pick the one that gives me the least extra noise. So we wanted to ask, can we capture the mouse's low power brain mode with tractable math, right? Pulling into this sort of gain density world where there's good theory, but also ground it in this pretty complicated biophysical process, right? So we bring our constraints to be some simplifying assumptions about the shapes of the tuning curve, but then we want to make our other constraints very biophysical. We want firing rate homeostasis, and we want to directly turn the knob that is the ATP budget. Okay? Yes, okay. So how do we do this? We have to turn those two biophysical needs into an expression of gain and density. I'm just going to go through this real quick to give you a flavor for it. The firing rate homeostasis, we say the rate normalized by the probability is going to be the gain over the density. What this means is that if I get wider, I have to also get shorter. The gain and the density change together. Okay, so this is what enforces the flattening of the tuning curve. Otherwise, if I get shorter, I drop my firing rate. If I get wider, I raise my firing rate. And then for the ATP budget, now we just have to say, okay, well, how much flattening should I do depending on how hungry I am, right? There's a good reason that we would want to tie this to the gain rather than the density, but you can justify whatever you want in the math. We're just going to say our energy is the expected gain function. All right, so this is just going to tell us how much are we flattening depending on how hungry we are. So this is what results in this field look like. It's kind of a mess. I want to focus less on what's going on at the party than who showed up. So at the top, we've got Infomax and Discremax, so we can handle a family of merit functions like we could before. And from there, we can predict stuff like the gain and density, which is going to tell us the widening, and we can also get out the Fisher information. Okay, so we're predicting all of the stuff that we would expect. And in the middle, there's a whole bunch going on. But what I want to just point out is that there's a couple of new faces. So we have this alpha, this E, this eta. Alpha is a hyperparameter. We're going to resolve it in a second. But E is an energy budget that we can actually connect to real numbers in ATP per second, which is the first time that that has ever been brought into one of these sort of simplified optimization problems. And this eta sub k is really exciting. It's going to tell us, okay, now that you've pulled down your energy budget, how much information have you sacrificed? What is the noise penalty associated with that energy budget? And additionally, it's going to depend on kind of this K is the overall activity level of the cell. There are orders of magnitude differences along the visual pathway and how many spikes get sent, and that's going to change some of your trade-offs and change what's optimal. Okay, so now we can take this this mouse data and we can sort of calibrate our model correctly, and we can predict everything from how much energy they saved to how much graceful failure to expect to how much widening to get, and we can get consistent with this mouse data while still plugging into, again, this family of analytical solutions in the literature. So we're pretty excited about that. Again, I'm throwing a lot at you. I just want to give you the flavor of what we did. So we start with this general framework, and we say, okay, we need to tie down the energy, add some homeostasis, we get our flattening. We do that by grounding those few new parameters in a biophysical model. What's cool about this is that we can now look at the other hypotheses in the literature, which previously were not really related to each other. They just made different assumptions. But now we can say these are a united family, and we can go along and basically say where these went off the rails. So the one that shortens, they mistook the firing rate for energy. a reasonable assumption, but one that's not borne out in the data. Oops. The one that widens, similarly, they mess up an exponent in how they estimate what the energy would be. Again, a reasonable hypothesis, but not what we see in the data. So now we can show that there's this whole family of models that are related to each other and can be grounded in biophysics. Okay, and in fact, our sort of gold standard here, we end up with the same mean firing rate, and they don't capture that behavior. Okay, how do we do this? Again, super high level. We just simulate a neuron. We have a few electrical parameters, and we just let them take on all the values. We figure out what happens in response to a single spike. We impose homeostasis. We get a noise energy trade-off. We impose our hypothesis that you're going to optimize your noise energy trade-off, and then we get this optimal decay path. Okay, so what this looks like in pictures, I'm going to let all of my parameters change. I'm going to get some amount of energy cost. Okay, and I'll draw out the parameter set that keep me homeostatic, and I'll get, in fact, the energy is pretty simple. It ends up having sort of this nice linear pattern. I can intersect that with my noise surface, and at each energy level, I just pick my lowest optimal noise, and as I get hungry, I'm going to move down this path. So we get a family of these curves, right? As we get hungry, we come down, but depending on if we have sort of a quiet cell or a very active cell, it'll look a little different. This is what lets us do these simplifications, but I think more exciting than the numbers that we got out of this is really the units on this plot. So I am directly estimating in ATP per second, but I'm also, my parameters now are the resting voltage of my cell and the leak conductance, right? How many ions are leaking across the membrane. In comparison to a tuning curve, these are much more accessible measurements. So to get a tuning curve, you have to have a microscope or an electrode in there. You have to have this mouse in a little tube. You have to show it these striped patterns. And then because it's noisy and we know it's even getting noisier as they get hungry, you have to repeat this experiment a whole bunch, and you have to do it for every single stimulus value. These are arduous experiments. This, you just need a voltmeter, right? It's a fancy voltmeter. It's not the same one from your physics lab, but anyone who can do any part of this experiment, they're already doing this as like an original step. This is a much simpler, more accessible way to do it. So if this model ends up being borne out by data, then we not only have like a cute mathematical model, but we also have a much easier electrical test to determine what state you're in and how your code might change as you get hungry or full. So just to zoom out, I know we skated over a whole lot of details. We've gone over the expectation. We care about what's in the world. We've talked about how there's different kinds of merit functions for different kinds of tasks. And now we've figured out how to tie these to real biophysical properties like the firing rate, homeostasis, and the actual ATP budget, how much energy you're really using. Okay, and not only can we capture a single population code, but we can actually capture an optimal decay that handles multiple states of that constraint. So this is our initial step into figuring out how both the goals and the constraints are shaping the information processing so that we might someday be able to understand these computational principles and pull them out into technology. That's important because of all of these, we're almost certainly the most expensive. Our brain runs on the same power as a light bulb, as opposed to, again, a giant data center. Additionally, all of these are going to fail gracefully as the available resources change. And I just think this is going to be super necessary for the next generation of vision technologies. So with that, I'd like to thank everyone who made this possible, and I'm happy to take questions. Yeah.
So you flashed up some G-Leak data. I presume you did so because it confirms or it agrees with your analysis, or
Was this, was-
right there?
oh.
Oh, we just, yeah,
I think it was actually this one.
right there. Yeah.
Yes. Okay. Yes. So basically, if you want to determine how much to flatten, you can either constrain the gain or the density, right? Because they vary together. We picked gain because there's a measured difference in the channel conductance. So which we think is analogous to gain.
I see. I see. Okay. A more philosophical question. So now attacks on computational functionalism, which is, perhaps long in the tooth, including from Hinton with mortal computation notions, et cetera, that once we get to the thermodynamic limit of computation, we no longer have substrate independence. We no longer have multiple realizability. And I'm just curious what your thoughts are, having looked at the instantiation in biology versus running this in code on powerful machines. What do you think is the future direction? Are we going to go to substrate dependence? Are we going to get rid of computational functionalism, and or what?
Yeah, I think at the moment we are so far away from being limited by the computational substrate. I think our algorithms are just deeply worse than the algorithms in the brain. And so I think eventually we might need to deal with that. But I think the current state, so right now in computer vision, the models are growing literally exponentially. It's a doubling size of about 10 months on computer vision machine learning models. We're going the wrong direction, right? So I think that could eventually be the question. I doubt we will ever get human level vision performance from the same voltage and wattage that we get in our brains, but we could at least get within an order of magnitude, and we're not even really trying right now. Yeah.
I was wondering, just like the whole data is from visual cortex, but just like the stimulation, just like coming first is the retina. What's the neural population activity in retina? Is there any just like future direction or just like existing direction, like compare just like the information in retina cell, there's also neurons, and compared to the visual cortex, what's the like a latent space differences, or just like what kind of information just like being emphasized or neglected in
That
visual
is a
cortex?
really great question. So one of the reasons that we wanted to have very different activity levels is that the retina fires about 100 times more than the cortex does. And so you'll have a really different trade-off and a really different state. And if you interpreted retinal data using a cortical model, you would almost certainly get it wrong. So yes, we're very excited about that. I think it's also likely that this information theory, in its simplicity and in its sort of non-robustness in some ways to recurrence, which is really key to understanding cortex, I think these theories might end up being much more useful in retina than in cortex. It's just that the data we scavenged that is currently available is from cortex. I'm very excited, actually. So I mentioned, oh yeah, these things are easy to measure. Our collaborator, Greg Schwartz, measures retinas all the time. So we're really hoping to actually run that experiment we proposed in mouse retina. It's a great question.
So I had a question about sort of part of, you started the talk with a sort of feedback loop.
Mhm.
I was wondering the extent to which this application of information theory to the biological system, is that inspiring and informing the biomedical community? Are they thinking differently about information and how the brain processes given the results that you've shown? In particular, one of the hypotheses was that the resting firing rate is increased under sort of low ATP availability. And was that an assumption that you had imposed, or is that something that's already well known in the biomedical community?
Okay, the piece of like what the low power mode looks like.
Yeah.
in the brain. Yeah. So that is my very simplified just so story of the Podamsey data that came out a few years ago. So there's basically one paper that asserts this change that ran the experiment. And we are trying to sort of put it into a functional computational context. We are not yet informing anyone of anything. I would love to see if this has some kind of implication for like a metabolic challenge. So diabetes in the retina is a pretty serious health problem that this could potentially connect to later. We've been thinking about it more in terms of graceful failures for technology. How should our camera design change? Also potentially, how should neural networks adapt? Right now, typically, if you want to reduce the power consumption of your neural network, you have a specific energy target and you retrain to that target. It would be great to have a flexible principle that lets you turn down the wattage on your neural network. But to do that, we would still have to understand better all of the factors that are playing into this. I don't think we fully understand the computational principle yet.
Yeah, and I just wanted to conclude by just mentioning, it seems like what you're proposing and what this other paper proposed is able to explain like hallucinations under like starvation, because if that's what's happening, then if the threshold's reduced, then you get a bunch of false firings, and those are going to, and it seems like that might also explain, you know, in terms of how different drugs might also induce certain activations of the brain, it may be also this threshold reduction. So it just seems to me a very interesting biological hypothesis that could inform a lot of drug development and inform a lot of treatments. And it seems like your analysis seems to sort of lend some legitimacy to that hypothesis. So I think it could be hopefully this can be communicated to the biomedical community in a way that they can appreciate it as well.
That's a very exciting vision. Thank you.
Thank you for the talk. And as you say, the biological brain is like really like use low energy and have all these advantages, and maybe this can inform the computer vision. And you also said the field of computer vision is kind of in a wrong direction. I just wonder, like, what do you think is the obstacles that prevent people use biological brain principles to guide the development of computational vision? Like, is it that we don't understand the brain yet, or is it a field gap, or yeah, I would appreciate your thoughts.
Yeah, oh, that's an excellent question. So yeah, I think part of it is that it is just very hard and not yet known, right? We worked on these problems for years and we got like a couple of maybe interesting in 10 years results, right? So if you want to like build a system now, we're nowhere near that. I will point out that the dominant AI technology is a simplified analog of a neuron, right? Like the primary algorithm that is driving most computing right now is something approximating or inspired by the brain. So I don't want to neglect that fact. But yeah, I just think it's difficult, and it's also the incentives of the field are very heavily drawn towards you make the benchmarks go up and then you win all the prizes. And typically the computation speed, the cost, the impact to the wider world is not included in any of our field's incentives, right? Part of that is because all of these properties that I said that they had, oh, they're compact, there's design diversity, they're fast or whatever. These are sort of hard to write down exact numbers for, which makes it hard to optimize towards. And it's also just in general, like the rewards of our larger society are reflected in the rewards of the field and amplified.
Just as a follow-up point to that, I think you make some great points, but like even in like the field today, there's still a lot of need for example, like low energy models, like models that can run locally on small devices and things like that. So what's preventing people who are looking for those low energy solutions to look towards bio-inspired like approaches?
Yeah. I mean, I think it's very difficult. It involves communicating across disciplinary boundaries, which is a lot harder than you might assume. I think some people are looking, and some people are sort of applying the problem, applying engineering tools directly to the problem on the shortest path. And I mean, to be clear, I think that diversity of perspectives, viewpoints, and approaches is important. But yeah, it's just a hard problem.
I would argue a little bit against that. So if I look at the problem, I see that the biggest issue is capitalism, right?
Well, once we fix that.
But let's think about it, right? You said one of the great things about natural systems is that they tend to fail gracefully, right? They can still perform, because it would be bad for any organism to be behaving as if it was completely healthy until they suddenly die. But if you want to keep selling people phones, it's actually really great if they fail miserably. So you are forced to get a new one. And if you want to overwhelm your competitors, it's also great that you make things more and more incredibly expensive. So I do not really think that it is because we cannot find the solutions or that people are not interested in the solutions. Is the entire system and the ones that make the rules that don't want those solutions to be taken and adopted. So
I think there can be more than one hard problem at a time.
There is pretty much in computer science one approach to these problems that is being accepted in all the conferences and pursued by all the big tech companies, right? And that is not about diversity. And it's not about letting people have choices. So I would say that that is really the biggest problem. And I would say that academics probably should resist that approach and try to do something different instead of just feeding stuff that makes the fat pigs fatter.
Well, I'm doing my part. Thank you.